The overall goal of this procedure is to study the remodeling of the endo-lysosomal system by intracellular bacteria using fluorescent nanoparticles as tracers. This is accomplished by first seeding HeLa cells in an 8-well chamber slide and incubating them overnight. The second step is to infect the cells with Salmonella enterica. Next, a pulse trace labeling of infected host cells is performed by incubation with gold BSA rhodamine nanoparticles. The final step is to observe alterations of the host cell endo-lysosomal system by confocal microscopy. Ultimately, quantification of the intracellular distribution of nanoparticles is performed using the Imerys software package.
The main advantage of this technique over existing methods, like immunofluorescence microscopy, is that even infected cells are analyzed.
This method can provide insight into the intracellular lifestyle of Salmonella enterica. However, it can also be applied to other intracellular pathogens such as Chlamydia species, Legionella pneumophila, or Mycobacterium tuberculosis. It may also be applied to other host cell types such as macrophage cell lines or primary cells.
The synthesis of 10 nanometer gold nanoparticles, or NPs, requires two solutions, solution A and solution B. To prepare solution A, add 2 milliliters of 1% aqueous gold chloride into 160 milliliters of milliQ water. Prepare solution B by adding 8 milliliters of 1% trisodium citrate dihydrate and 160 microliters of 1% tannic acid into 32 milliliters of milliQ water.